Hello, and thank you for joining today's webinar. I'm Mark Stewart, Vice President of Science Policy here at Friends of Cancer Research, and I'm thrilled to welcome you to this important discussion. This webinar serves as a follow-up to our recent meeting, Advancing the Future of Diagnostics and Regulatory Innovations, which took place on February 4th. That event brought together experts across the field to explore how emerging technologies such as AI-enabled diagnostics and digital pathology are shaping the future of precision medicine. We also examined critical challenges in developing diagnostic tests for rare biomarkers and the need for regulatory flexibility to ensure these innovations reach patients. Today, we'll be diving into key takeaways from that meeting with a special focus on what these developments mean for patients and the advocacy community. We recognize that patient perspectives are essential in shaping these conversations, and this webinar is really an opportunity to ensure that advocates are informed, engaged, and empowered to take action in advancing diagnostic innovation. We have a great panel lined up, moderated by Doyle from Eli Lilly and Company, who will lead us through the discussion. And I encourage all of you to participate. There will be a dedicated Q&A session in the latter half of the webinar, so please feel free to submit your questions throughout the session. And with that, I'll hand things over to Megan to introduce our panelists and kick off the discussion. Megan, over to you.
All right. Well, thank you very much, Mark, for having me, and to Friends for hosting this webinar and bringing us all together. Thrilled to be here and to be hosting this esteemed panel. We have with us George Green from GA Green Consulting, LLC. George is an independent consultant working with the pharma and biotech industries to develop precision medicine and companion diagnostic products. We also have Joan Mancuso, a patient advocate serving as a Friends advisory advocate, emphasizing the importance of accurate diagnostics based on personal experiences. And we also have Alain from Tempus AI, where he is the Senior Director of Regulatory Affairs. So thank you all for joining us. Let's jump right in. So in terms of our discussion last week about recent advances in diagnostic testing, I'd love to hear from you what excites you about the potential for the recent advances in diagnostic testing. And maybe, Joan, you could kick us off from the patient perspective about what excites you.
Thank you, Megan. And also I want to thank Friends of Cancer Research for developing this project and also for involving advocates in it right from the beginning. I had a personal experience with HER2 positive breast cancer back in 2003 when I was diagnosed with stage 2. My slides were looked at twice by 2 different institutions. The first result was HER2 positive 3+, and the second result a couple of years later was HER2 positive borderline, which is completely different. At the time, I learned that that's not so uncommon. Also, AI project that can improve accuracy. As you know, AI is in the news all the time now. At the San Antonio Breast Cancer Symposium, there were 2 sessions on AI with a lot of presentations. AI has been involved in cancer imaging for a while, not only with mammographic screening, but also with, for example, looking closer at brain metastases, like with the FDA-approved AI tool, vBrain, which can kind of see much smaller lesions of roughly 2 millimeters, does better accuracy at measuring them, and also looking at potential heterogeneity changes. And that's an aid to radiologists for brain MRIs. So in general, there's been a lot going on in the AI arena. And so I think that this is excellent to be involved in this. But thank you.
Thank you so much, Joan. And so building on that, I'd like to talk a little bit about how AI-enabled technologies are transforming diagnostic testing in oncology. George, maybe you could give us some thoughts on that.
Yeah, no, of course. And again, as everybody, thank you very much to friends for the opportunity to participate in this. This is these experiences are great from both sides of the camera. Um, yeah, so AI is really evolving in many spaces in the diagnostic industry. Everything from using it as a basic research tool to generate hypotheses that we can then test to find better ways to manage patients, to treat patients, all the way to just very practical applications in the lab with approved tests. And I think, largely the focus of this project that, that was presented at the last meeting was focusing on HER2, which has been established for a very long time, but there's always opportunity to improve that. And Joan's story is an excellent example of that. Using AI and digital imaging tools allows us to much more accurately and reproducibly get test results for patients, which then of course leads to better patient management. And I think that's going to be really critical going forward as the treatment options become more and more defined by the patient's status, and and obviously as those patients go through their journeys, we need to have better tools and more effective tools to manage those patients. I also think, Joan's story was around the accuracy of the result. There's another piece of this story too, which is that in the current healthcare environment, pathology is a very specialized skill, and pathologists are becoming more and more overloaded as far as work they need to do. And by simply having tools like digital image analysis, we can enable them to focus on the important and more difficult to interpret results and allow basically a computer to do the initial workups on patients so that they don't have to spend the time doing it, and they can just rather than doing all that work, they can confirm the results. So we get both the benefits of efficiency and accuracy out of these tools.
Thanks, George. And Alain, being from a company that does AI, what can you say about how AI-enabled technologies are transforming diagnostic testing in oncology?
Yeah, thanks, Megan. I think that's a great question. And, one thing I want to emphasize is that, AI has made big inroads already into medical device space in general. If you look at the number of devices that have been authorized by FDA to date that incorporate some kind of AI, it's almost 1,000. But only a small percentage of those, maybe like 5 to 10% ballpark, are in oncology, and most of those are in radiology. So something like the digital path work that Friends has been trying to advance is really kind of like the next frontier. And what I think we're seeing here is this leading edge of AI being applied as a tool in developing diagnostic tests in oncology specifically. And I think one of the places that it may provide a lot of advantage is somewhere we can see parallels in other fields like cardiology, where AI-based software is being used to extract kind of hidden information that is not otherwise available to clinicians from standard cardiology tests like ECGs and ultrasounds. And that's information that's just being left on the table, and AI is able to kind of pull that to the forefront. And so I think work like that is really what we're going to find moving into oncology diagnostic testing. And then we'll see, like George talked about, these approaches being incorporated into clinical workflows, and then the broader medical ecosystem. And then what I'd love to see after that is kind of real world measurements of impacts on public health. And I think we're going to see that.
That's great. Thank you so much. Yeah, we talked a lot last week about how AI and digital pathology could advance diagnostics in this space and patient identification potentially someday in oncology. And I guess one of the things we also talked about that I'd like to transition to now is we also discussed, considerations for validating companion diagnostics and other diagnostics for rare biomarkers, and how we might need regulatory flexibilities in doing that. And I think that's reflective of another set of advancements in oncology, which is targeted oncology therapies that, as we learn more and more about the drivers of cancer, we're identifying actionable biomarkers for these rare subsets of cancers, or that may be a key driver present in a small population of cancer patients and that could potentially respond to novel treatments. So as these treatments become increasingly tailored, we also need diagnostic tests to identify the patients that might respond to that treatment. And as we can talk a little bit, I'd like to talk a little bit about this and sort of how these advancements in targeted therapies are driving this need for more diagnostics for rare biomarkers. And then we can talk a little bit about, why we need to validate the assays that detect them. George, I don't know if you have anything to add from the precision medicine space and working in that space about how targeted therapies are really driving this need for diagnostics for rare biomarkers.
Well, no, absolutely. And that's, I think for any therapy today, that's really the goal, is to identify the patients that are going to best respond to that therapy, rather than just taking a shot in the dark as we did, 20 years ago. And I think it's continuing to evolve. Many of the therapies, and if you look at for example, the immuno-oncology therapies today, you use a biomarker test that is relatively useful but not perfect. And that's likely because they're looking at a very simplistic view of what's available, the information, to Alain's point, the information that's available in that pathology slide. They're looking at just one or two elements of that, where there's probably hundreds of elements that can tell you more about the story for that particular patient under those particular circumstances. And being able to use AI to interpret that is really the next step in this. Because right now, what we do today is we take a single piece, a single data point, and make a single decision. Whereas what we're dealing with, cancer is a disease of an environment, and you have to really address the environment, not just one single data point within that environment.
Yeah, definitely. And I think what I'd add to that is that in addition to sort of AI-enabled tools, even just developing your sort of more standard assays or diagnostics for rare biomarkers, as we develop these new targeted therapies for these rare subsets of cancer, we will need a companion diagnostic to identify them, the appropriate patients. And to develop those tests, we have multiple steps, which we talked a little bit about last week. Analytical validation, which are studies to identify the test performance characteristics, how it performs, and clinical validation, studies to evaluate whether the test identified the right patients, those who will respond to the therapy. But I think the precursor to our conversation last week, and we'll delve into this a little bit more as we talk about impact to patients, is how, especially for these rare biomarkers and indications, there are challenges with validating these tests, such as getting sufficient samples or well-characterized samples to perform these studies. And that was really a key part, I think, of what we discussed last week in terms of that piece. How do we drive that validation forward, and what regulatory flexibilities are needed so that patients can truly take advantage of these advancements in targeted oncology therapies. And with that, I'd like to talk a little bit more about impact for patients. What all these advances we're discussing today really mean for patients with cancer. How do they have the potential to improve care and patient outcomes? And Joan, I was hoping you could speak to that and share your perspective on that.
Thank you, Megan. I was thinking basically what you, first of all, I have to say that the HER2 project gave me a great appreciation for the challenges that scientists face in trying to get this to work. I also agree with improving outcomes, but improving outcomes for patients where the treatment works and where the companion diagnostic is accurate as to who should have that treatment. I myself was concerned in the HER2 digital pathology project about whether or not there was any information gathered about race and ethnicity, for example, because that could potentially affect who would benefit as to how accurate the diagnostic is. I learned then there were over 1,000 slides, but they came from one hospital in Belgium. And it just turns out Belgium is not allowed to collect information about race and ethnicity. And after that, I began to find out that, some countries are allowed to collect that information and others are not. So I was just like somewhat, disappointed that we wouldn't even be able to have that, rather to see whether or not there was any association with any of the levels of HER2 positivity or HER2 low, whether there would be anything there. On that note, a lot has been coming to the forefront about race and ethnicity in regard to efficacy of particular drugs as well as side effects. I had mentioned that Dr. Melissa Davis was a scientist at Morehouse School of Medicine. Did the plenary session at San Antonio Breast Cancer Symposium, where she was trying to untangle the socioeconomic factors from potential biologic factors. So she noted a few things, such as the use of hormone treatments in breast cancer, and how Black women did not really have as good an outcome as white women have had with that. Also, she cited Dr. Rachel Martini's study in Africa of looking at sub-Saharan African groups on the east and the west coast of Africa, and also ethnic groups there, and how, like in the United States, they have a higher probability of having triple-negative breast cancer. Dr. Martini was also able to discover an immunologic profile that is different. So these factors can, in fact, affect whether patients respond. So it could go deeper into race and ethnicity. Now, a best case scenario would be that it wouldn't matter, that the diagnostic works for everyone. I mean, that would be the best way to go, but sometimes that's not the case. And this also occurs, let's say, in Asian populations, where a retrospective study looked at like chemotherapy, PARP inhibitors, CDK4/6 inhibitors, and other treatments, and how Asians respond to treatment. So the study showed that in most cases, Asians respond the same as non-Asian populations, except for in the case of tamoxifen, where it was noticed that they don't respond as well, but also that they may respond better than non-Asian populations with CDK4/6 inhibitors. This also extends to side effects as well. So, for example, Black American women are known to, who have taken trastuzumab, have known to have twice the rate of heart failure related to trastuzumab, and that was discovered or noted in a study that was done at Memorial Sloan Kettering Cancer Center. There's also other things too, with Asian populations can have higher toxicities, hematological toxicities, including neutropenia. So I think that the whole point is that to not overtreat patients where they're not going to have the benefit, but also to be able to identify which patients are going to benefit from treatments, and also which patients that a diagnostic assay actually represents. And so that's some of the things that I think are very important going forward and seem to be gathering steam.
That's so helpful. Great. Thank you, Joan. And I'd like to maybe turn to George to weigh in on this as well, in terms of what are the immediate impacts to patients versus more long-term regarding some of these advancements we're making in diagnostics, both on the digital pathology and the rare biomarker side.
Yeah, I mean, I think with any new technology that moves into the diagnostic space, there's always a lag in its adoption as both the technology gets proven, as well as obviously there's regulatory and health economic factors that tend to either slow it down or speed it up, should usually slow it down. And I think, if we look at, especially, just taking digital pathology as a use case, there's obvious immediate utility for it. And we do see some products on the market now where what we're doing is more or less assisting the pathologist. As I mentioned earlier, the pathologists are overworked, and there's a number of products out there now that are just helping the pathologists make those decisions and report a result. And so those are starting to emerge and build up momentum in the field. Really, the implementation of standalone products, or products that are actually even going beyond a standalone product that's just doing what the pathologist did, but a standalone product that would be interpreting a pathology image with features that couldn't be seen by the human eye. And that's really the ultimate goal of this, is to get that additional information. Those products are probably a significant way down the road, both because of our inability to establish the efficacy benefits of those, unless we do clinical trials that focus on that, the regulatory barriers, because there's no established regulatory process for those yet. And then a very practical aspect of this, which is you have to have physicians who are going to use the test. You're going to have health insurers that are going to pay for the test. And it's unfortunate, but in this field you have to follow the money. And these products do get adopted as they become a part of the healthcare system. They don't just get adopted because they're cool technology.
Right, and to Joan's point, they'll be adopted in the interest of patients, actually serving the patients.
Yes.
And making sure that once we know they can identify the right patients and identify the right patients for therapy, that will drive that clinical adoption that you're
Right.
speaking of.
And it's a bit of a circular thing. You have to prove that before they'll adopt it, but they have to adopt it before they'll prove it. So it is, and that tends to be why diagnostic products have a long ramp up period
Right.
in this.
Right, exactly. So I'd like to move now into some of the things we discussed last week about sort of challenges in diagnostic testing generally, and things that we touched on in sort of the Digital Path project and the Rare Biomarkers and Diagnostics project. So maybe, Alain, you can kick us off with giving us your thoughts on sort of some of the key challenges that still exist in these areas.
Yeah, I think that some of the key challenges that came out of the work that's been done in these collaborative groups are, I think, things that we already knew about. I mean, I could just go back to Joan's example about getting different pathologists to review the same information and giving different results. And that's something that we saw with the software too, that you give the same slide to 6 or 7 different pieces of software, and sometimes they disagree. A lot of times they do agree at the cases that are kind of the highest and lowest scoring, but in the middle there's disagreement, and you see kind of the same patterns for pathologists. So I think that's this one kind of consistency challenge that is going to be something that needs to get solved. I think another one that really was highlighted is just something that George mentioned, which is this kind of efficiency challenge. If you look at taking 1,000 slides and getting those scored by individual pathologists, well, I'm ballparking like 2 to 5 minutes a slide. You're looking at hundreds of hours or dozens to hundreds of hours to score those, where you could take 1,000 images and run them through an algorithm and have them scored in 5 minutes. And then I think that really brings us to kind of the third challenge, which is I think what's going to be most helpful in moving this forward, is how to combine those things, how to get the efficiency benefits from the software and combine those with pathologists to get kind of both, optimized for efficiency and accuracy.
That's great. And George, I don't know if you have anything to add there in terms of kind of the key challenges that still exist beyond maybe what you've mentioned earlier.
I think Alain touched on it very nicely. I do think that I think that, and I'll just reiterate, getting the complexity, the complex ideas, the complex interpretations established in the absence of prospective clinical data is going to be a big issue. What I would add, though, is that I think what we did see from this particular project, I was very pleasantly surprised by the degree of alignment we saw between, north of half a dozen different algorithms that were completely independently developed by companies who compete with each other, and yet they all ended up pretty much agreeing. That's really encouraging for AI and the approaches that we're taking. It says that, again, you take the human out of the factor and everything's going to converge on truth. I hope.
Right. And in terms of sort of the validating test for rare biomarkers or indications, I think we still have some challenges there that we tried to address in the white paper, but that will persist. And I think that's why it kind of led us to the conclusion that we need some regulatory flexibilities there. As I mentioned earlier, there's these 2 steps in validating a companion diagnostic for a rare biomarker. And availability of clinical samples to conduct those sets of validation studies can be really challenging. So I would add that that's still a challenge we're trying to address. You know, sponsors may often enroll on local tests that already exist for the patient to reduce wait times for patients trying to enroll and maybe enable more patient-centric trials of a new targeted therapy and avoid rebiopsy, but that can exhaust samples because the samples used in the clinical care and management of the patient. So therefore, we don't have necessarily always leftover samples to develop a diagnostic test. I think we also face ethical considerations that contribute to this challenge. You know, we have, as sponsors, a lot of stakeholders that we're trying to manage who may be resistant, again, to subjecting patients to an additional biopsy for the purposes of gaining a sample to validate a test, particularly if there's no other treatment options. You know, a provider may want a patient to be able to enroll in a trial quickly and not want to delay that for an additional biopsy. And similarly, I think we have challenges in that we often as a sponsor have to develop sometimes even more than one companion diagnostic for a given rare biomarker or indication, maybe a liquid biopsy and a tissue biopsy-based test, so there's options for patients, or for different geographies when we're developing targeted oncology therapies. Our development is usually global, and so we may need a different companion diagnostic for each geography based on what platforms and technologies are available there. So all of that contributes, I think, to this ongoing challenge that we tried to address in the white paper of availability of clinical samples. How do we conduct that validation? And I think led us to the conclusion that we certainly need to partner with regulators and have some regulatory flexibilities there in terms of how to validate these tests so that we can get them to patients as soon as practicable. And I believe we're getting ready to turn to the Q&A. So I think at this point, I will move us in that direction. So I believe we have a question that's already been submitted regarding the digital pathology project, and noting that digital pathology generates massive amounts of data. So how can we help manage and process this data efficiently while ensuring patient privacy and compliance with, of course, applicable laws and regulations such as HIPAA or the GDPR, and particularly when we're developing AI models. So I don't know, Alain, if you could maybe speak to that just as our sort of resident AI developer.
Yeah, sure, Megan. I think one thing that Tempus does when working with patient data, I think it's probably just a general practice, is de-identifying the data for kind of research or development purposes. And then you can maintain kind of a key that's kind of independently held of the folks that are doing the development that can be used to kind of re-identify those if needed. But you know, all the information that is you know, kind of protected health information is removed from the samples and data that's being used for the development and testing. And so I think that is kind of, I mean, I think it's a great point that there is tons of data, but there are some good ways, I think, to try to help ensure patient privacy and protection of that data when doing the work with the samples.
If I could maybe add on
Go
this,
ahead,
just because
George, please.
it's probably it's a soft spot or whatever for people, but you know, there are, over the last couple decades that or more, unfortunately, I've been in this business, there's been tremendous advances in the checks and balances around this. There are guidelines from the regulatory agencies around how you can use human source specimens. There are investigational review boards that are very sophisticated now at asking questions about how they're going to be used and making sure they're being properly used. And even at the level of diagnostic or pharmaceutical companies, there's many internal processes such as de-identification, segregation of data away from, you know, different groups within the organization, and how you handle the data after it's been analyzed. That I really, you know, my confidence in our ability to preserve privacy is pretty high, especially compared to what things were, you know, again, you know, 20, 30 years ago when all this started. It's a lot of thought is put into it, and people, it's hard to reassure people that their private information is safe, but I think everybody has the same sort of view and perspectives on this, that it should be.
That's great. Thank you so much. Surely, yes, we've developed a lot of systems in place, to your point, George, to address the changing regulations. You know, we have state privacy laws being passed all the time, in addition to some of these things in sort of at the national level or at the international level. And I think, to your point, companies work really hard to quickly respond and establish systems to comply with those laws and regulations. So another, oh, go ahead, Joan. Were you going to add something?
Oh, yeah, no, I just want to add that patient anonymity is very important. And I feel somewhat also confident that it's gotten to the point where it's pretty well protected. So I just wanted to note that. Thank you.
That's great. And so another question I'd like to chat a little bit about, we talked about in the rare biomarker situation, we're developing tests, how we sometimes face a shortage of available clinical samples to do some of these validation tests. So FDA recently published a guidance document on considerations for including biopsies of tissue, you know, of the actual cancer in the organ itself in clinical trials. And I'm wondering what your perspectives are in kind of balancing the ethical considerations around, you know, requiring tissue biopsies in the clinical trial with the potential contributions to research that these biopsies could provide. And I'd love to start with you, Joan, just from the patient perspective, to get your thoughts on this as a patient.
Sometimes a biopsy is required in a clinical trial. It may, the trial requirements may say that it, had to be within the last 6 months, let's say, for example. Also, this would be, of course, again, tissue biopsy, not a liquid biopsy. So I feel that it's important to be, first of all, that might be a requirement of the trial, just in general, to have that biopsy in order to just verify that the patient has the diagnostic or the mutation or the alteration that's being looked at. So I think it's important to have the guidelines to make sure that everything is in order with taking the biopsy. And even though it's a burden on the patient, I think in the area of metastatic breast cancer, like I have metastatic breast cancer, that sometimes the trade-off is worth it. So even though it's a burden, patients might be really, open to doing it to be able to advance, advance treatments if possible.
Yeah, that's a really good point, Joan. And I think the guidance notes that sort of trade-off question that, if it's a tissue site that poses higher risk, alternative approaches to the biopsy could be considered, balanced by, if we need to do that biopsy, having a strong scientific justification for sort of the critical need for that. So to your point, it's sort of that balancing of risk versus potential contribution or criticality to the research. I don't know, George or Alain, if you have something to add.
Yeah, I'd just love to also add in the importance of kind of consent and communication with the patient and, talking with patients about what the purpose of the biopsy is. And, I think, Joan, to your point, I think a lot of patients may be, willing to provide this not only for the trial purposes, but for other research purposes, but they should really understand what the purpose of the biopsy is and why they're being asked to undergo that procedure. And, I think we can give patients a lot of credit here that they are, sophisticated in instead of being able to understand that information if it's communicated in the right way and make a good decision.
Yeah, and George, do you have anything to add?
Sure, yeah. A lot. So this is something I discuss with especially biotechs in the early stage space all the time, because the importance of having current and then potentially even sequential biopsies post-treatment to understanding the translational medicine story around, what is the drug doing? What is the best way to select patients? It's extremely powerful to have those samples to do that. But on the other hand, it is about patient safety first and consent first. And so the vast majority of the time we have a discussion around is it required for the patient to be a part of this clinical study? If it is, we give them the option to participate. They can get a biopsy, but we would never require them to. If there's follow-on biopsies, we make we explain the case for why we're doing it. We, for the most part, try to capture it within the routine standard of care. Joan, you mentioned follow-on biopsies sometimes are necessary just to manage the patient. So we try to incorporate into that. And really the last resort is going in and saying, on week 7, we want you to come in for a biopsy because it's mandatory. That's very undesirable, but in some cases, to do drug development, that may be what's necessary. And so you just do it with the appropriate boundaries.
Yeah, and I would just add from our experience with precision oncology trials, that again, we have to really think about the risk it presents to patients, as you just noted, and whether or not requiring that additional biopsy. So for example, maybe just for purposes of central testing in a trial with a prototype test, is that really necessary? Because in our experience, it can also lead to significant delays in getting patients on trial and potentially initiating the investigational therapy due to extended wait times for the results. So we have to balance all of these things. And I think that's one of the things we addressed, I think, really well in the paper is when you're going through this balancing and you realize that it's not, there are many reasons why you may not be able to obtain additional biopsies or samples, or it wouldn't be patient-centric to do so. What are the options that we might have for validating the test? What are the regulatory flexibilities we might want to discuss with regulators so that we can continue to develop the test in a really robust and expedited way, but without posing any additional risk to patient safety? And it looks like we have another question. So someone had asked whether there's any avenue to use samples, possibly from a deceased patient who died of the very rare cancer, or if they had permission or consent from their next of kin. I will give that to maybe Alain from the scientific side, or George. I don't know that that's always possible, but welcome your thoughts.
Yeah, that's a great question, really interesting, I think, especially in the context of the rare biomarker work that we talked about in last week's meeting. And honestly, I don't actually know the answer to that, and it may be a function of consent, and I don't know if it's actually possible. George, I don't know if you have any direct experience in this area.
Yeah, I was just going to add, George, I could see, if you had appropriate consent, maybe the samples could be added to a sample bank or used in analytical validation studies. I think in clinical validation studies, we typically need the clinical outcomes data from the patient. Did they actually respond to the therapy? So presuming that the patient has deceased, unless you have that clinical outcomes data, I think it would be challenging to use for that purpose. But welcome your thoughts too, George.
Yeah, no, I totally agree. I think there's the sample banking, sample acquisition for development of diagnostics is also a ongoing discussion, and very challenging at times, especially when you get into rare biomarkers. The use of banked specimens in general is extremely valuable when you're doing your analytical validations, as you mentioned, Megan. And we source a lot of them for those purposes, as well as for obviously early development and research purposes. But you do need clinical efficacy, I think, to validate a precision medicine indication for a product. I mean, that being said, sourcing from deceased patients, I have seen it tried in the past. The problem tends to be that the quality of the specimen, the consents, everything else. I mean, we're interrogating what should be a living patient, a living organism, and as soon as death occurs, unfortunately, a lot of these processes stop or change, and you just don't know what you're going to get. But we have done it in some cases where it's really the only option.
Thank you. And Joan, did you have something to add?
Yes, I would like to add that now in breast cancer, we have sort of what's developed is known as the rapid autopsy programs. I know there are several places where this is being developed, and one is at University of Pittsburgh. So the idea would be that on the patient's death, the patient has already indicated that they would like to, let's say, give some of their organs for scientific purposes. But the problem is that the autopsy has to be done very rapidly. The family has to know about it, and there has to be coordination, let's say, with the program, let's say, at University of Pittsburgh. So this is coming along. This is something, at least in breast cancer, that has been moving along and developing in different places, and it's particularly for the reason that the person noted, that suppose somebody is deceased, and and they want to give their tissue for cancer research, and of course that would have to go to the family member. Maybe it would be in a person's power of attorney, their medical power of attorney, or something of that sort. But I did want to mention that those programs are starting to pop up here and there, and are being developed.
That's super helpful, Joan. I wasn't aware of that program. That's great. And we have another question from the audience in terms of asking how do we standardize advanced biomarker testing and other forms of diagnostic testing across both academic and community center settings. And I think that the idea being, to provide access to patients and reduce financial toxicity of costs associated with testing. So ensuring all patients cover the cost of these tests. And the question is really pointing out that advanced technology and testing leads to improved personalized medicine, but only if patients have access without a dramatically increased financial burden. I can just say from my perspective, I think this is an ongoing challenge in oncology. We have clinical practice guidelines that suggest using NGS testing, for example, and advanced biomarker testing to identify patients who may be eligible for a targeted therapy. But sometimes getting everyone to actually embrace those guidelines and do that testing can be challenging. I've had challenges with that, even in my own experience caring for a patient with cancer at academic centers, having to sort of advocate for that testing to be done in accordance with clinical guidelines. So I think it's an ongoing challenge that I do think sponsors, other stakeholders, thought leaders, friends are trying to address in many different ways. I don't know, Alain, George, or Joan, if any of you have additional thoughts on that.
Well, I would say if you look at, I completely agree with your comments. And what I would add is that it's either good or bad, but as technology continues to evolve and improve, one of the challenges we have is that we develop a test, it gets reduced to practice, it gets distributed to the big centers first, and then the smaller centers, and then there's almost immediately a next evolution of that product that does it even better yet. Mm-hmm. And a great example would be next generation sequencing testing, which is a tremendous tool for managing oncology patients. Basic oncology panels have been around now for, depending on how you count it, 5 to 15 years. And honestly, I think the costs and the use of them has largely been commoditized for the basic panels within the last 5 or so years. But now what's happening is there's additional information to be had. Now they're sequencing things beyond just mutations, and they're going to many more things around the methylome and RNA expression that are giving us more information, but also adding to the cost and creating, again, access issues. So I think part of this is as things get adopted, they become accessible, but it does take some time for that to happen.
Yeah. I agree completely. Joan, did you have something to add?
Well, we often feel like maybe the more information, the better, but sometimes it's not useful as of yet. So for research purposes, it's a good idea, but it takes a while for maybe it to get translated, into the clinic, of course. And that used to be a big excuse sometimes from oncologists if you were asking to have like a genomic test or sonomic test or a germline test. Your oncologist might answer you So what would I do with that information? Would it change your treatment tomorrow? And in which cases, in many cases, it wouldn't. But it's really good to, I think, to continue to gather that information for use down the road.
And I think the other part of this, beyond just patient access to the tests, is then being able to kind of collect and centralize and access that information that comes out of it, because sometimes the results of the test are going to be just kept at one local site, and it's really hard for people to go in and get access to that. And then you're trying to collect results from tests that have been done across the country. And right now it's, some of that information is just fragmented. And so ways to think about being able to have better access or centralized access or disseminate that information is also going to be an important corollary to getting the tests out.
Yeah, agree with all of those points. And maybe while we're waiting for some more Q&A questions from the audience, we can talk about what needs to be done to address these challenges that we've discussed and accelerate progress. How can advocates, including maybe some of those on the call, play a meaningful role in advancing the innovation we're seeing in diagnostics? I'd love everyone's thoughts on that. Maybe, Joan, we could start from your perspective again as a patient advocate yourself.
Thank you, Megan. I feel that it's very important to keep reminding clinicians and scientists about what we feel is important. This has happened, for example, in cardio-oncology, right? Like at first we didn't really hear much about it. I actually have been affected. It's hard to tell in my family. I have heart failure, but it's not the type that I got for taking Herceptin from 2007 to 2020. Because it runs in my family. But the more and more it is showing up more and more conferences where there are sessions on cardio-oncology, for example. So that would be one way. There's also another, which is the right dose. That is something that we keep hearing more and more about. First, it wasn't too much. We would hear it here and there. Now it's been brought up again, and it's really appearing in conferences. So it's very important to keep in front of people about this. Another way to get involved is to be involved in projects, whether it's grant proposals for laboratory science, or whether it's for clinical trials, and that it's important to get involved on the ground floor where you can bring up the questions in the laboratory of what you think are important. For example, I was mentioning about race and ethnicity, and the same thing for clinical trials. If you get involved on the ground floor at the beginning of the development of the trial, you can also bring in your thoughts. So for example, the TBCRC, the Translational Breast Cancer Research Consortium, I was with them for about 3 years with University of Pennsylvania, where they come with an idea. So scientist comes with an idea and the advocate is involved right from the beginning. All the advocates involved right from the idea and how it gets taken all the way through as to actually becoming a clinical trial. And sometimes you could see a session at a conference or a poster at a conference that has the TBCRC number on it, right? Because that's where it's originated from. So I think it's important for advocates to get involved on the ground floor in these projects in order to let our thoughts be known about what we think is important to study from the patient perspective.
Thank you, Joan. And I don't know, George and Alain, if one of you wants to weigh in on kind of what you think we need to do to address these challenges and how advocates can help. Maybe we'll start with you, George.
I was afraid you were going to do that. It's rare diseases especially are tough because you're dealing with a fairly small patient population and a challenging both to develop the drug and the precision medicine strategy, as well as then to do the clinical validation of that. And I do think that one of the things probably the pharmas can do better and the advocacy groups is working together, pick a project, pick a rare disease and work together to both include, like FDA, include health authorities in the dialogue about how are we going to do something with this. Even payers, how are we going to pay for this? Because we can't develop a $2 million per dose test or drug. But the reality is, in these smaller, rare diseases, they can be very challenging to develop, to validate, and to implement. So I think working more closely together would be a really big assist on that stuff. And I have seen that actually in some other space. I have a cardiovascular program right now that's actually got some great advocacy involvement, and it's a super benefit for it.
And any thoughts from your perspective, Alain? I have some as well, but want to make sure we get your thoughts.
Yeah, I mean, I think what George just touched on, the idea of like collaboration and crosstalk amongst all the people involved is really what's needed. I just think of, from a very small kind of microcosm of that, the digital pathology work, that collaboration that Friends spearheaded here, that you can see the power of getting different groups together to work on the same problem. This idea that you take the same image, you analyze it with 7 pieces of software, and when you get different results, then you can get those people together and ask why did that happen? And that is really powerful because it provides insights that individual developers wouldn't have come to on their own. And I think that can be expanded to kind of the broader ecosystem of, patients, advocates, developers, providers, payers, to get people working together and collaborating to solve these problems.
Yeah, I agree with all that's been said about getting involved in some of these multi-stakeholder collaborative efforts. It really helps, I think, all of us to hear the patient perspective. I think in terms of some of these challenges we've discussed, for example, in like the rare biomarkers or diagnostics setting, it's really important for drug developers that we get patient input in sort of study design. A lot of drug developers are trying to do patient-focused drug development, and having insights like some of those that Joan shared earlier about biopsy, and having insights from patients into the design of the study and the challenges associated maybe with follow-on biopsies can be really important, and I think really helpful in overcoming these challenges and finding that right balance. I also think, as we mentioned in the white paper, we certainly need these regulatory flexibilities that we pointed to in the white paper to help us develop and validate these tests in, a robust way, but as soon as practicable so we can get them to patients. And I think we identified some really reasonable suggestions and ways we could engage with regulators to, gain regulatory flexibilities that enable us to develop a test where it's scientifically appropriate.
Can I wake Mark up for a minute? I was going to say, maybe there's an opportunity here for Friends to broker some of those relationships. If Friends had a database of advocates who would be interested in particular rare diseases, and the pharma companies was aware of it, and we could marry projects together, it might be a great way for us to get that information.
Yeah, and I think we have one more question. I do know there are some organizations that do that can help connect us sponsors to patients to give input on trial design, and it's very, very helpful. I think we have one more question. So in the current climate and context, how do we make sure that patient voices and needs are at the center of the conversation and the center of the decision-making table and aren't ignored? So I'd love to hear, again, maybe I'd like to hear your thoughts, Joan, based on your experience as a patient advocate, and then we can all add our experiences.
Based on my own personal experience, I got involved in advocacy in 2010. So that's 15 years ago. And patients were pretty much ignored, and metastatic breast cancer, which I was diagnosed in 2007, was also really ignored. There were hardly any sessions, or at conferences, if there were any, they were the last hours of the last day. So I want to mention that over time, we have gotten more involved in projects, and clinicians and scientists have been paying a lot more attention to our thoughts. And it's been difficult over time, but it is getting better and better. Now, hopefully, we will not have a reverse of course in the current context. For example, like I was discussing about maybe the importance of race and ethnicity in determining efficacy with treatment, as well as potentially more serious side effects. So I would hope that we would be able to continue, along that lines, and that won't become, more difficult for us. In other words, that gains that we've made in getting our voices heard in some areas, will not be reversed in the current climate. I also suggest that advocates or patients that are interested in getting involved kind of hook up with a patient support a organization, because oftentimes the support organizations are very helpful in pushing out information and learning about information and allowing advocates to get involved in individual projects. So if you're very interested in becoming active as an advocate, that's clearly a way to go.
That's great and super helpful, Joan. And I can say, from the sponsor perspective, I think one way is to bake some of this into your processes, bake into your drug development processes where you're going to get patient input, where you're going to get those kinds of insights. And I do know sponsors do that in their patient-focused drug development programs, but I think it's always a good question, as we received from the person on the webinar, to say, okay, how do we make sure those aren't being ignored? And I think one way is to try to bake it into the process. Any other thoughts, George or Alain, before we move to concluding remarks?
Yeah, I know Mark's gonna come on in a moment here. If I would just say one thing, it is that, so I moved from the diagnostic industry, pure diagnostics, into the pharmaceutical industry about 15 years ago. One of the things that really struck me when I made that move was that the vast majority of the people involved in the pharmaceutical industry are focused on patients. It may not be clearly stated all the time, but there's a very common mission that we want to help people. And it is what there are people who have spent 20 years developing one drug, and it has been very important for them to do that. And I do think it's reassuring to me that that's there. The underlying premise is there. Sometimes it's not always visible, but it is there.
Thanks, George. And with that, I think we'll turn it over to Mark for some concluding remarks.
Thank you so much, Megan, Alain, Joan, George. That was a very insightful discussion. And I also want to thank all of our participants today for joining us. I hope this conversation provided valuable takeaways on the evolving landscape of diagnostic innovation and opportunities to shape its future. At Friend, we're committed to continuing these discussions and working collaboratively to ensure that cutting-edge diagnostic advancements translate into real-world benefits for patients. If you have any additional questions or feedback, please don't hesitate to reach out. We welcome your engagement. With that, we'll wrap up today's session. Thank you again for being part of this conversation, and we look forward to staying connected.